Thank you, guys. Okay, the lesson is recording now. All right. So I know I've been experiencing, I think, a little bit of lagging between the screen and my device and all of that. It actually turns out that I needed to increase my Wi-Fi speed. I think that's what it's called. So I've increased it. I think from next week, the lessons are going to be smooth because apparently once you are on Teams and having a lot of people joining your Teams meeting, I don't know, it takes a lot of whatever. They explained it. So that's why there is a bit of lagging, and sometimes my pen doesn't write properly because everything becomes slow. So I think from next week it should be smooth because I called them today and they said it will take 72 hours to upgrade my speed. Okay, so let's speak about this. Just a little bit of introduction on protein synthesis. So I think the word protein is quite common even in everyday language. Because we speak about food and then we speak about protein shake or the amount of protein that people who go to the gym need to take and all of that. So this word to us is very common, right? Protein. Now, yes, our body makes proteins, but it's not enough to sustain every little thing that is needed by proteins. So we then need to consume some proteins to obviously add to that. So how our bodies make the protein, it will use DNA. So we are going to use DNA in order to make specific proteins that are needed by our bodies. Now, these proteins are needed to build our cells. These proteins are needed to build tissues, which eventually build the different organs that you have in your body. Now, to also understand how important proteins are, they are also needed to make enzymes, which are basically biological catalysts. So, are substances that speed up reactions in our bodies. And another one is to make hormones. Hormones are chemical messengers. So they are obviously secreted by different organs in our bodies, and that is directed by the brain. So if there's a shortage of whatever, the brain is going to stimulate a certain organ or certain structure to release a hormone. That hormone will be transmitted or transported, rather not transported via the blood and go to do whatever. So we basically send messages through hormones. So all of those things that I've mentioned, they're actually made from proteins. Hence, our bodies need to make the proteins first. So if you eat an egg, you are actually eating a protein. If you eat meat, you're actually also eating a protein, which will be built in your body. Okay, the basics out. We understand why this is important. Now, what I want us to note here is our goal in protein synthesis. The word synthesis means to make. So our goal is to make the protein, but we're not going to make protein out of nowhere. We are going to use the genetic code, which is the sequence of bases in DNA. Once we speak about the genetic code, you need to know that we are speaking about DNA. So in order for you to make a cake, you're going to need flour and this and this and that, right? To follow those steps for you to build or make a cake. You don't build a cake, you make a cake. So for us to make the proteins in our bodies, we are going to start here. We are going to use the genetic code. Now, don't stress when someone says genetic code and then you get confused. What exactly are they speaking about? They are speaking about the sequence of the nitrogenous bases in the DNA. You see that A, C, T, G? There's actually a sequence there which can be a repetition of the bases or whatever. It can be A, G, T, T, A, C, whatever. That sequence will determine a particular gene, or that sequence will determine a particular protein that needs to be formed. So a genetic code is the sequence of the bases. Right. So we are going to use the genetic code, a particular code of the DNA in order for us to make a protein. But before we make a protein, we will need to first make different RNAs. Now, in our introduction, we spoke about 3 types of RNA. We spoke about a messenger RNA, we spoke about a transfer RNA, and the ribosomal RNA. Now, when we use the genetic code of DNA, we are going to use it to first form the messenger RNA. The first type of RNA that we're going to form is the messenger RNA. And then from the messenger RNA, we will be able to form the other type of RNA, which we call a transfer RNA. So we use DNA in order for us to form two different types of transfer RNA. From the transfer RNA, we are then going to get a chain of amino acids. So I've spoken about this before. A chain of amino acids, and amino acids are building blocks of proteins. Now, once you form a chain of amino acids, minimum 50, they then form the protein. So from here, we are going to then form a chain. AA is amino acids, not alcohol, anonymous, or something else. It's amino acids. Then those amino acids are going to be bonded together by peptide bonds, and then will form the protein that is required. Now, if I then make this example and I say, okay, in this circle, that's an amino acid, and this line is showing the peptide bond line. So that's an amino acid, and another amino acid, and another amino acid, and another amino acid, and another amino acid. That is going to form a long chain. That long chain of amino acids is referred to as a protein. But where did it all start? It started by using the genetic code for DNA. Remember the rules, guys. Once I have my notes here and I start writing, I can't see you on the other side. I can't read the chats. I can't do anything. So the minute you have a question, unmute and then ask me a question. As soon as you unmute and you start speaking, I'm going to stop and raise my hand to show that I'm paying attention. Okay, just to explain the genetic code, this is the sequence of bases. I've spoken about it. What is that sequence? T, C, A, G, G, the order of the bases. Now, DNA is divided into sections, which are called genes. This word is important. As much as it's mentioned in this topic, it's not really explained in detail. Once we look at meiosis and genetics, we are going to look at it in detail, and it is a very important terminology. Now, if we take it back to maybe the first or the second lesson, I spoke about the chromatin network, which carries the DNA, and we find the chromatin network in the nucleus. Now, just before cell division, this chromatin network is going to condense and form these thread-like structures, which we call chromosomes. So the chromosomes carry the DNA, chromatin network carries the DNA. The only difference is the chromatin network is just an entanglement of the DNA. And then when it becomes condensed, we then call it a chromosome. Now, I think in another lesson, I will speak about the chromosome in detail. Now, within a chromosome, and this whole structure carries DNA, there'll be different sections in that chromosome. So just like in a house, there'll be different sections of the house. There's the kitchen, there is the dining room, there's this and that. In the chromosome, there'll be different sections. There can be a section here, and a section here, and a section there, and wherever. Now, sections in a chromosome that code for a certain characteristic are called a gene. So meaning a section in a chromosome is called a gene. This section here is called a gene. And that section codes for a particular characteristic. That characteristic can be my eye color. So meaning the sequence of bases that we find in this section determines my eye color. The section over here, meaning the sequence of bases that we find in this section, may determine another characteristic, which could be my hair texture or my blood group or whatever. Now, these genes are very important because they carry information that will enable us to make different types of proteins that are needed by the cell. So already all of this is controlled by the brain and the nucleus itself. So in order for the protein to be formed, that will be guided by the brain to say, okay, this person's body needs this type of protein. This type of body or this person is needing this type of protein, and then the cell is going to produce that. I was just reading the messages. That's why I was I'd like to get a recording too. Okay. Now, the only difference between the DNA of a human and that of an organism is basically the sequence of bases. So all organisms will contain their DNA. The only difference is the sequence of those bases. In organism one, you can have TTACC. In another organism is ACCTG. And remember, this DNA is very long, super long. Now, the sequence of the bases that we are going to look at will determine what protein will be made. So the sequence of the bases for whatever section that we are going to use is going to determine the protein that is going to be formed. And I think at this lesson, at this level in grade 12, we just doing protein synthesis and finding out the protein that is coded for, that is formed, nothing more. So not necessarily the information about that protein or where exactly in the cell it's going to be used and all of that. So we don't look at it. Okay, then in terms of the DNA that you have coding DNA and non-coding DNA. So coding DNA is the DNA that will code for the protein. So you only have 2% of the DNA, meaning from the 100% of DNA that you have, there will only be about 2% of that will code for making proteins. So that means when we make the proteins, we are going to speak about DNA unwinding. So it will be a section of DNA that codes for a protein that is going to unwind. It won't be just any other section. Now, we do also have DNA that controls what proteins are made and when, but it's referred to as non-coding DNA, and that makes up about 98%. So it doesn't code for a particular protein, but it controls what proteins are made and when. Yeah, they won't ask you much about it, but just the background. Okay, let's find out more about these proteins. The definition is this one. Protein synthesis. What is this process? This is a process by which proteins are made in each cell of an organism. And we are making these proteins in order to form enzymes. I spoke about enzymes, in order to form hormones and new structures for cells. So in order for your cells to function, they will need certain structures. So when there is a lack of those structures or a shortage, we need to then make the proteins. Now, just like when we spoke about a block and a house for DNA, and we said the building block for DNA is the nucleotides, and then those nucleotides together will form a house. Same with proteins. So proteins will be that big house. So protein will be this big house. There is the house. But in order for us to make this house, which is protein, we need the building blocks. I've mentioned this already. Now, the building blocks of proteins are amino acid, and the complex molecule itself is a protein. So in order for us to make proteins, we need amino acids. Now, I've already spoken about it again. So if you have an amino acid and another amino acid and another amino acid and another amino acid, remember with the blocks, you need to join the blocks together, and you normally use the cement and other things to join the blocks together to make a house. So with the amino acids, we are going to bond them together by these bonds here. We call them peptide bonds. Peptide, I think it's a D. Yes, not a T. Peptide bonds. We need to remind ourselves, when we spoke about the nucleic acids, the bonds that we spoke about there were the hydrogen bonds. So this is supposed to be hydrogen, right? Then when we speak about proteins, the bonds between the amino acids are called peptide bonds. So let's not mess this one up. Now, in terms of the amino acids, because now you're thinking, oh, okay, we are going to use amino acids in order for us to make proteins, but we actually only have 20 amino acids. We are going to use these 20 amino acids and combine them in different combinations to form long chains that will form a protein. So from these 20 amino acids, we can actually form a protein that has about 300 amino acids. Now the question would be, okay, how do we move from 20 to 300? Because they will be combined differently, and the same amino acids can be repeated in that sequence. So if you're having a proline, it doesn't mean you'll have a proline and another one and different ones. You can have a proline and then a repeated proline and then in arginine, a proline again, trypopine, alanine, blah, 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 blah. From the 20 that we have, they may be repeated in order for us to form a long chain. Now, the number of amino acids and the sequence of amino acids determine the type of protein. So the protein that is going to be formed will be determined by the number of amino acids that are bonded together and the actual sequence of that amino acid, actual sequence, yes, of the amino acids. So what do we have here? It's our building blocks. The block is the amino acid. That's the monomer, right? Then the bond between the amino acids, that's the peptide bonds. We'll leave it at that for the slide. Remember, ask me a question anytime, guys. Now I'm going to explain this, but in a grade 12 syllabus, they don't break it down like this, and they don't test it like this. So the terminology, it's not really assessed, but when you have two amino acids that are joined together, we call that dipeptide bond. The word di means two. So if you're just having two amino acids joined together, that's not a protein. We call it dipeptide. Then, if you are having more than 3 amino acids but less than 50 that are bonded together, we call it poly. Poly means many. The word poly means many. So polypeptide, that means you have more than 3 or from 3, but less than 50, meaning 49. And this one, we're calling it a polypeptide. Again, we can't call this one a protein. We will only call a chain of amino acid a protein once it has 50 or more amino acids. So if it's just 2 amino acids, it's a dipeptide. If it has 3, 4, 10, 20, 43, 49, it's a polypeptide. Then once it has 50 amino acids bonded together, it's a protein. But like I said, these two terminologies are not really assessed or talked about in a grade 12 syllabus. In a test, they will speak about a protein being formed, but you are not going to see 50 amino acids. It's not possible. You will see 3 or 5 or 6, whatever number. They will just tell you that is a section of the amino acids that they're giving you. So you cannot expect a chain of 50 or 300 amino acids. You take what you are given. If they say we are using these amino acids, even if they gave you 3, you take what they're saying. These two terminologies also not assessed that much, but at dehydration synthesis, we are basically forming a complex molecule. So we are moving from the amino acids or the monomers to form the house. So we're using the blocks to form a house. Then hydrolysis, we're breaking down the complex molecule. So we're breaking down a house to then form our building blocks. That's all it means. So one side we are building, the other side we are breaking. Let's get to the juice. Protein synthesis. Now, the big question would be, where will this take place? Man, we're speaking about proteins being formed and all of that. The whole process is taking place within a cell. The whole process. Now, there's 2 stages or phases within this process. The first phase is going to take place in the nucleus, and the second phase is going to take place in the cytoplasm. Let's break it down. The first phase is going to take place in the nucleus, and then the second phase or second stage will take place in the cytoplasm. Remember, these are structures of the cell. So if you look at the diagram that I've projected over here, this is a cell. And in this cell, that is where protein synthesis is going to take place. Now, what you're able to see on your slide is this structure over here, which is the nucleus. So that means the first stage, which we call transcription, it's here in yellow, will take place within the nucleus. And then the second stage, I said it's taking place in the cytoplasm. There it is. Maybe I need to change my color to red so that it can be visible. There it is. There's translation. It will take place in this jelly-like substance over here where metabolic reactions will take place in the cytoplasm. That's all that we need to know for now. Now, this slide is very important. I spoke about protein synthesis being the process, and this process occurs in 2 stages or steps, but sometimes they also call them processes themselves. So you want to make a cake, you first need to mix. Step 1, you mix the flour, you mix it with everything else. Step 2, I think you put everything in the oven. I told you guys I can't bake, I can't cook. Well, not too bad, but yeah. I think that's how you do it. So it's the same thing with this. The goal is to build the protein, but we need to start somewhere. The first stage of protein synthesis is called transcription. So stage 1. This is what needs to take place first. Stage 2 or step 2, that is translation. So if we are following steps, step 1 needs to take place before step 2. You can't have step 2 taking place before step 1. That's not how it works. So this stage will need to take place first before step 2 or stage 2 can take place. Let's look at this. Now, the most important thing to note here is that with transcription, which is the first stage, we are going to use DNA to form a certain type of RNA, which is messenger RNA. We are going to use the genetic code. Remember, I spoke about a genetic code in order for us to form messenger RNA. Where is this taking place in terms of location? It is taking place in the nucleus. There is the nucleus. There is the process. Now, this can also be confused with DNA replication, and I'm going to do it in this lesson so that we don't confuse it at all. Now, when you have this, you can cross it out. They're not going to ask you about it, the type of enzyme. So we have an enzyme that will help carry out the reaction here to control what is happening, what is being formed, where is it going. We don't need to know the name. We just need to know there's an enzyme that is going to control all of that. Then the second stage is translation. Once the messenger RNA is formed, there it is over here. Remember I said step 1 needs to take place before step 2. Now, once the messenger RNA is formed, we are going to use that to form a chain of amino acids. And once we form the chain of amino acids, remember, if you have a minimum of 50 amino acids, you then have your protein. Now, where did it all start? It started in transcription. And the location of this particular process, that is the cytoplasm, there it is in this green part over here, that's the cytoplasm. So what are you taking here? 2 stages, transcription, translation is the second one. Location, nucleus, and then the cytoplasm. Now, I can tell you with confidence that some of the learners that find protein synthesis challenging will, however, remember the names of the stages. They will remember that there's a transcription and then there's a translation. The problem then comes when they have to remember between transcription and translation, which stage comes first and which one is second, because these words look and sound the same. So we are going to work it out together. I'm going to change my color. Let me see if I can erase first because I want to use this part. Oh, my pen is behaving today. I wonder what happened. Okay. Okay. Ooh. I shouldn't have said anything about the pen. Okay. So remember I said there's very high chances that you'll remember that there's transcription and translation, but then forget which process comes first and which one is the second one. I want us to look at the first 5 letters. I think it's 4 or 5 letters. The first 5 letters for both stages are the same. What do we have here? Trans and also trans. Those first 5 letters for both processes are the same. But now let's look at the letters that come after trans, after S. Because in a test, you can write them both down and then you follow this strategy. If we look at the letters that come after trans, in transcription, the letter is C, and then in translation, the letter is L. There is L over there. Then you think about it. In the alphabetical order, which letter comes first between C and L? The letter is C. So that means since C comes first before L, the stage with C, transcription, is the one that comes first. That's how you need to remember it. So trans, then C, L. Which letter comes first? C. So that means the one with the letter C is the first stage, and the one with the letter L is the second stage. All right. Now let's get into it. This is the business. You need to know how to describe this, but there's 6 points there. So make a note with the slide. It's a copy and paste, 6 marks. You need to know how to describe that. But let me come here. Now, let's remind ourselves about this process. Transcription, that is the first process. And in terms of location, it will take place in the nucleus. That's where it's going to take place. But this process is very similar to DNA replication. The first 2 points here are the same. The section of DNA double helix coding for proteins unwind. If you take it back to DNA replication, we said the same thing. We said the DNA double helix is going to unwind. The only difference here is this part, of which is not important. Even if you say the whole sentence without saying coding for proteins, you'll still get a mark. Let's look at the second point. Weak hydrogen bonds between the nitrogenous bases of DNA and double-stranded DNA unzips to form 2 separate strands. The bonds are going to break, and then the molecule unzips to form 2 separate strands. These 2 points, 100% the same to the points that we mentioned when we did DNA replication. Now, very easy to confuse the two. So we are going to redo DNA replication before we do transcription so that we can see exactly how they are different. Point number 3 says one strand is used as a template to form messenger RNA. I have a question. So if you know the answer, please do unmute and give me the answer. Point number 3 says one strand is used as a template to form messenger RNA. What was different in point number 3? Because this sounds very similar to DNA replication. What did we say in DNA replication at point number 3? Unmute and tell me. What did we say? Anyone?
In DNA, we said both strands, they serve as a template, or each strand serves as a template.
Thank you so much. I will tell you, I've been marking paper 2 for more than 10 years. Learners confuse that, but you are in this lesson. You're not going to confuse that. In DNA replication, we spoke about both strands being used as templates. In transcription, we are going to speak about only one strand being used from the 2. Let me not waste time. Give me a second. I'm going to get an empty page for us to do this. So if you have a page on your side, please, let's do it together because this can be confusing, but give me a second. Okay, there you go. Just give me a second. I'm trying to get a page. It's on my screen, but it's very slow to set up the way I want it to be set up. Okay. I think I'm winning now. Okay, in 5 seconds, you should be able to see a clean Word document. Please do thumbs up once you can see it on your side. No. Okay, I need to see a few thumbs. Okay, cool. Happy, happy, happy. Okay, let's start. I'm excited. Let's do this. Okay, I'm just waiting for my screen to come to the party on my side. Okay, so we are firstly going to remind ourselves of DNA replication before we speak about transcription. Now, if we remind ourselves of DNA replication, let me just, I hope I won't disturb this, but I want a thinner pen. Come on. Okay, so when we spoke about DNA replication, remember the main goal of DNA replication. If you can't see my pen, if you feel like it's too thin, just unmute and tell me you can't see it or something, right? Okay, so the main goal of DNA replication was to make a copy of DNA. That was the main goal. And where did all of this take place? In terms of location, it took place in the nucleus. That's what we know, right? And we find the nucleus in the cell. So this is the nucleus. This is everything that we did already last week. I'm going to move very quickly from it. But now I am having this big landscape page so that on one side, we can look at DNA replication, and then on the other side, we can look at transcription, which is very important. Now, if we remind ourselves of what happened last week in DNA replication. So this was happening in a cell. First point, the DNA double helix is going to unwind and the weak hydrogen bonds are going to break and the molecule will unzip and form two separate strands. These, are the two separate strands. Now we then said both strands, that's what Luyanda just said also now. Both strands are going to be used as a template. So both strands will then be called template strands. So since we are using this one as a template, we'll refer to it as a template strand. Since we are also using this one as a template strand, we'll refer to it as a template strand. We can also refer to it as a coding strand. Template, but sometimes in an exam they call it coding. So this one can also be called coding strand. The reason we are saying template, we're going to use it. The reason we are saying coding, we're going to use that code, right? So 3 points done. DNA double helix unwinds, hydrogen bonds break, the molecule unzips, can still be considered second point, and then both strands are going to be used as templates. Now, if these are the bases that we have on our strands, I'm going to be quick. If you can't see anything or there's something I'm messing up, unmute and tell me. So let's say this side we have TAGCTA. Okay, meaning the bases that we are going to have on the other strand, since DNA is double-stranded, remember? So the only reason we are having these two separate strands is because the bonds in between are now broken. So, but initially these two were paired together. So that means this side will have an A. If I mess it up, please do correct me. It's been a long day. A, then T, then C, then G, A, and then T. Yay, we're happy, right? But now point number 4 says, we are going to use both of these strands to then form new strands. But how are we going to form these new strands? Now, free DNA nucleotides. Ooh, I need to make sure I mention this. What are we going to use here? We are using free DNA nucleotides. Because remember, we are forming a DNA molecule. A molecule of DNA will be formed by DNA nucleotides. Remember, a nucleotide will have that phosphate, that sugar, and then a base. That's what we are going to use here. So how will this work out? We explained it last week. A strand is going to be formed and an enzyme is going to control this to ensure that the perfect pair is the one that is going to complement the exposed bases. So these bases over here and here are the exposed bases. So now the one that will be paired with T will obviously be A. So a free adenine base is going to be paired with that available T, then here a T, then here a G, a C, then a G, then an A, then a T. Same business on the other side. So this will be, yo, I'm so happy my pen is behaving today. So this will be a T, an A, a G, a C, a T, and an A. This was DNA replication. We used this strand and that strand. And then at the end, we double checked. Did we achieve our goal? How many nucleic acids do we have? We have in 1 and 2. This will be a DNA and this will be another DNA. And then we checked. Did we make an identical DNA molecule? Yes, we did. What's making it identical? The sequence of bases are the same. This is TA, TAA, TATGC. You get the business. We leave it at that. Now, at the end of DNA replication, now, okay, let me forget about it. Covered it last week. Okay, so DNA replication done. Now I want us to look at transcription. Transcription. Now, what is the goal here? Anyone unmute and tell me what the goal is. I need the information about the goal and the location. Remember, we are comparing this to DNA replication. Anyone unmute? And I don't want the same person speaking for 2 hours. All of us are here, and I want all of us to be able to speak, please. Anyone? And I'm not moving up until I get an answer. I need you to tell me what the goal is. What do we want to achieve in transcription? And where exactly does it take place? Oh, well. Rosego, what's the goal in transcription? What are we making here? Anyone, guys, please? Please? Anyone?
Isn't it the formation of messenger RNA?
Thank you so much. Thank you so much. That is our goal. Here in transcription, we are forming messenger RNA. Guys, if y'all are quiet, I'm also going to be quiet. I'm not going to move up until I get an answer because this is straightforward. I want us to get this. And I've mentioned the goal already. I've mentioned the location. So if we're going to waste time, I will also waste the time. I'm just going to wait. This is a lesson. It's about me and you. It's not just me speaking for 2 hours. That's not learning then. It becomes a very boring lesson. Okay? Then I need someone else to tell me about location. Where will this take place?
In the
Where
nucleus.
exactly in the cell will it take place? I'm hearing someone.
In the nucleus, ma'am.
Thank you so much. Inside the nucleus. Both these processes are taking place inside the nucleus. That's why they are easily confused. This is our nucleus. Right? Then, what's the first thing? The DNA double helix is going to unwind. The same point as DNA replication. The weak hydrogen bonds are going to break. And then the molecule unzips to form 2 separate strands. These are the 2 separate strands. And remember, these 2 strands are DNA strands. Both of them. Now let's write bases, DNA bases. I'm just going to include 6 bases here. 1, 2, 3, 4, 5, 6. I'm trying to match the position. Then maybe we get a T, G, A, C, C, T. Okay, that means on the other side, we'll have A. Remember, if I'm making a mistake with this, I mean, I'm repeating it 100 times a day, so I could be making a mistake. Just let me know. A, C, T, G, G, A. We're still happy? These are our DNA strands. I want us to remind ourselves, these two strands are DNA strands. Now, the third point, this is where transcription becomes different from DNA replication. The third point, we are only going to use one strand from the two strands. We are going to use one strand in order to form, our goal is to form messenger RNA. Now, the question would be, okay, from the two strands, which strand are we going to use? Now, for me and you, we are going to use the left strand. That is going to be our strand that we use. The question would be, okay, what about in a test? How will we know which strand to use? They are going to tell you. How will they tell you? By labeling the strands. Now, the strand that we use will be called a template strand. I can't go back there. The strand that we are going to use will be called a template strand. We're using what? Using it to form messenger RNA. The strand that we are not going to use to form messenger RNA will be called the non-template strand, or will be called the non-coding strand. The one that we use to form messenger RNA will be called a template strand or a coding strand. So between the two DNAs. But let's look at DNA replication. Both our strands were template because we used both of them. Okay, let me not waste time. So one strand is going to be used as a template to form messenger RNA. How are we going to form this messenger RNA? We are going to use free nucleotides. But which nucleotides are we going to use? We're not going to use DNA nucleotides. We are going to use free RNA nucleotides. And what is the reason for that? We are forming an RNA molecule. So we need nucleotides that contain a ribose sugar. We need one of the nucleotides to be a uracil. So now free RNA nucleotides are going to be used here to form a strand. So a base that pairs with T is the one that is going to be available here to be paired or bonded with T. So I'm going to draw this line to show that we are forming a strand. Now I need you to unmute and help me out here. I do need you to unmute and help me out. So from the free nucleotides, remember we're focusing on a base. So which base is going to pair with T? Please do help me out, guys. Please do unmute. There's quite a few people in this meeting, and I don't want the same people to be speaking. So please do unmute and let's work on this. So which base from the four? And remember, since we are forming messenger RNA, we are going to use A, C, G, and and U. Okay, so T will be paired with which base? Which base is going to pair itself here?
With you.
Will it be U?
A.
Will it be A? Is it A or U, guys?
You. It's A. You. It's you.
Okay. Okay, let's look at this. That's why I need you guys to be active. That's exactly what I need so that I can address mistakes. If you're quiet, I won't pick up any mistakes. When we have the four bases on RNA, we follow the same rule. A becomes paired with U, not T becomes paired with U. Remember, in the bases of RNA, we do have A. So in this case, as much as we are forming a messenger RNA molecule, A is available, and this A will pair with the U in an RNA to RNA molecule. But now we are pairing an RNA molecule with a DNA base. So we cannot put U here because T doesn't pair with U. T will still continue to pair with A. Remember, T does not pair with U. U replaces T. So this is where it can become confusing. So you're not going to pair T, as much as there's a T in DNA, we're not pairing it with the U. We can't pair them together. They are double ringed bases. So A is available and A perfectly still pairs with T. Okay, I hope that is clear. If anything is unclear, please do stop me. Then which base is going to pair here? The second one?
C.
Thank you. Remember, I'm not moving. No answer, no movement, guys. Then A, which base are we going to pair A with?
U.
It's you.
Thank you. Then C will be paired with?
G.
Thank you. Then C will be paired with G. Then I need the last base. T will be paired with?
A.
A.
Thank you so much. Point number 4, done. We used free RNA nucleotides to form a messenger RNA strand. How did we form this messenger RNA strand? A will be paired with U. If we have an A in DNA, it will be paired with U. C will be paired with G. But if we are pairing a T that is in DNA, we still pair it with an A because A is available and it will be paired with the T. Now, then what happens after this process? Now, this messenger RNA gets formed. Now, once the messenger RNA strand is formed, it's going to leave the nucleus. Now, how will it leave the nucleus? From the first lesson, if you remember, the membrane of the nucleus was like this, right? And there were spaces in between the lines to represent the nuclear pores. So this messenger RNA strand, after it becomes formed, it's actually going to leave the nucleus through the nuclear pores and then go and attach itself to the ribosome. It will then go and attach itself to the ribosome. But where does transcription ends? Transcription ends as soon as the messenger RNA is formed and it leaves the nucleus. We are done with transcription, but not done with protein synthesis. But before I continue and speak about translation, I'm happy to take a question. 1 or 2 questions. You should have a question, guys. Please, 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 please. You should have a question. Anything that you found confusing or anything that you need clarity on, please do ask me now. Yes, Tabo? Or is it Luyanda? I'm seeing a few mics. Yes, I'm listening. Okay, no questions. All right. So as soon as the messenger RNA, there is our messenger RNA, there it is, it leaves the nucleus. It doesn't leave the cell. There is the nucleus over here. It leaves the nucleus and then in the cytoplasm, it finds the ribosome. I'm going to represent this in the ribosome. So how am I going to do it? Let's say this is the cell because I'm running out of space. This is the cell and this is the nucleus. This is where transcription took place. We had our 2 strands and then our messenger RNA got formed there. It leaves the nucleus and then finds itself in the cytoplasm. It attaches itself to the ribosome. Now I need the exact bases that we had here. So it's ACU. Okay, come on. There you go. I'm trying to change my color to this. Come on. There you go. So these are the bases, A, C, U. Where am I getting my ACU? The bases from this very same messenger RNA strand that we formed, GGA. Okay, oh, my strand is too long. Okay, I see. Sorry, guys. Okay. So I think we're having G, U. All right. GGA. Just going up and down is killing me. There you go, GGA. And this is our messenger RNA. This messenger RNA is now attached to this organelle in the cytoplasm, which we call ribosome. Now, at this stage of protein synthesis, this is now translation. We are done with transcription. We made our messenger RNA, it moved out of the nucleus, done with transcription. Now, the messenger RNA, the same one that was made in the nucleus, it has now attached itself to the ribosome. This is where the second phase is going to take place, which is translation. All of this is taking place in the cytoplasm. So location is the cytoplasm for this particular stage. The ribosome is found in the cytoplasm. Now, what are we going to have here? In the cytoplasm, there's another type of RNA, which is called transfer RNA. Now, this transfer RNA has a specific shape. It's normally represented as like a fork-like shape, but it's something that looks like this most of the time. Oh, I'm messing it up, but yeah, it looks like that. And you have a number of transfer RNAs in the cytoplasm. Now, what you do need to know is that in a transfer RNA, at the bottom of it, it is going to have what we call, let me not give you the terminology. Let me ask you guys, who can remind us, what do we call 3 bases in a messenger RNA strand? What do we call the 3 bases? We are having 3 bases here and then 3 bases there. What do we call? I'm going to call out people's names now. 3 bases in a messenger RNA strand. Kamu? Kamu? 3 bases in a messenger RNA strand. What do we call them? Kanyisa? What do we call the 3 bases in a messenger RNA strand?
Ma'am, don't you call them codons?
Perfect. Thank you. Codons. That is very important. Now, this is a codon, 3 bases, 1, 2, 3 bases in a messenger RNA strand. This is another codon. So that means in the diagram here, we are having 2 codons. I'm bringing up this terminology because it becomes important when we speak about transfer RNA. In a transfer RNA, the base of the transfer RNA, this section over here, is going to carry anticodons. Now, an anticodon is complementary to a codon. What do I mean by this? These are the bases of a codon, meaning a transfer RNA that is going to come over here is then going to carry, or the anticodons are going to be determined by the codons over here. So how do we form this? We make it sound like a pairing of bases. A will be paired with what? With a U. C will be paired with a G. U will be paired with an A. These 3 bases here are anticodons. So a codon, or rather an anticodon, is complementary to a codon. How do we form an anticodon? You think of, or you basically pair the bases of that codon in order to get the anticodon. Now, one side is the anticodon, and on the other side, it will carry an amino acid. Remember the amino acid, that building block of a protein? Exactly that. But It doesn't carry any amino acid. It will carry a specific amino acid. Now, how will it know which amino acid to carry? It will be determined by the codon. So it will go, oh, okay, in the codon, this particular codon 1, I'm going to label them like this, codon 2. Codon 1, the bases are A, C, U. Okay, that means the anticodon there will be UGA. Now, in the cell, in the cytoplasm, there are amino acids that are just floating there in the cytoplasm, different amino acids. Now, what the anticodon or rather the transfer RNA is going to do, this is a transfer RNA. What is it going to do? It will then move around the cytoplasm to collect a specific amino acid, an amino acid that is coded for by this particular codon or anticodon. So if amino acid X, I'm going to label it as amino acid X. There's no such in biology, but that's how I'm labeling it. So if amino acid X is coded for by ACU or UGA, this is the same thing, guys. Codon becomes complementary or anticodon becomes complementary to codon. This transfer RNA will actually move around the cytoplasm or wherever it is to go and collect this amino acid and bring it over here. That's why I said that a transfer RNA doesn't carry any amino acid. It will become specific. So amino acid X is the one that is brought to the ribosome. So where is it? There is amino acid X. There it is. I'm trying to change my color because it's becoming all green now. Okay. This is amino acid X. Same thing happens to the second codon. Oh, okay. We are having GGA. So that means the anticodon will be CCU. No T business here. We are moving from an RNA to another RNA, messenger RNA to a transfer RNA. So what will it do? Okay, now CCU, I need an amino acid that is coded for by CCU. Maybe amino acid Y is the one that is coded for. It will then come back carrying that amino acid, bring it over here, and that will be amino acid Y. So what do you then have? You have amino acid X bonded to amino acid Y, bonded to amino acid Z, bonded to amino acid X, bonded to whatever amino acid. Now the chain of amino acid is formed. We get 50 amino acids. That is our protein. Done. What is the function of the transfer RNA? Is to bring a specific amino acid to the ribosome. That amino acid is coded for by the codons. How many bases code for an amino acid? 3 bases. So an amino acid will be coded for by 3 bases. So 3 bases make up 1 amino acid. I'm happy to take questions before I go back to the slides. This is the beginning and the end of protein synthesis. I'm happy to take questions. Okay. Let me go back to the slides. I'm going to stop sharing the Word document and then go back to sharing the notes. Get ready to start speaking, guys. I will be asking a few questions. Okay. Let's start here again. Transcription. The section of DNA double helix unwinds. That's a mark. I think I didn't ask you guys if you can see my screen. Can you see my screen? I'm back to the notes.
Yes.
I need some. Okay, thank you so much. Oh, I love responses, guys. Thank you so much. Okay, then weak hydrogen bonds break between the nitrogenous bases of DNA and the double-stranded DNA unzips to form 2 separate strands. In 1 year, if you are lucky, you can actually get 3 or 2 marks from that sentence. Depends. 1 strand is going to be used. Please make a cross next to this word and write both. So you can remind yourself that if you write both wrong, if you write each strand wrong, you saw that we only used one strand. One strand is going to be used as a template to form messenger RNA strand. The DNA template is used to form complementary strand of the messenger RNA. There's no point there. This is done by using free nucleotides. Where are we finding these nucleotides? In the nucleoplasm, not in the cytoplasm. You make a note that we are finding them in the nucleoplasm and not the cytoplasm. This is for a mark. The mRNA is complementary to the DNA. mRNA now has a coded message for protein. That's a mark. And then the rest of the points, no mark for them. They are just explaining how the process takes place. mRNA formed from DNA, it will detach itself from the coding strand. Then 3 adjacent nitrogenous bases of the messenger RNA strand are called codons. These code for a particular amino acid. Messenger RNA moves from the nucleus to the cytoplasm and attaches to the ribosome. Now the question would be, oh, okay, what about that section of DNA that unwinded at the beginning? After messenger RNA leaves the nucleus, it goes back to its natural shape, becomes double helix. Okay, this is what happens. There's the DNA. Only a particular section coding for protein is going to unwind. Can you see that the rest of the DNA did not unwind? It was just that section. A messenger RNA strand is formed, and then it will move out of the nucleus to the cytoplasm. There is transcription still. Now, even here, you are able to see which strand was used as a template. So this is the messenger RNA being formed. You can see that we used this strand. That was the coding or the template strand. Now, these are the exam guidelines. This is what you need to write in order for you to get that 6 marks. Even if you use the other slide, it will still be the same. The double helix DNA unwinds, 1 mark. The double-stranded DNA unzips. I normally say start with this point first because the bonds will need to break before the molecule unzips. Okay, then 1 strand is used as a template to form messenger RNA using free RNA nucleotides from the nucleoplasm. Please make a cross and write DNA nucleotides. We did not use DNA nucleotides. We used DNA nucleotides when we did DNA replication. Messenger RNA is complementary to the DNA. Messenger RNA now has a coded message for protein synthesis. There is no mark for that. You are just telling us that messenger RNA moves out of the nucleus to the cytoplasm and goes to attach itself to the ribosome. Okay. This is for you now. Review the DNA sequence below and work out the sequences of messenger RNA that will be produced during transcription. This is the DNA molecule that you are given. What are we asking from you? We are asking you to form a strand of messenger RNA. I will give you 3 minutes. That's a lot of bases. I will give you 3 minutes for you to form your messenger RNA strand. 3 minutes, nothing more. Please don't be too quiet, guys, please. I've been begging you for more than an hour. Please, please, please. So 3 minutes. 1 minute left. Are you guys done? Please do respond. Say no, yes, something. Are you done? Can we mark? Not done?
Done.
Done. Okay. Everyone else, are we done? Can we mark? Yes, no? Do you need 1 extra minute, 2 minutes?
1 more minute, ma'am.
Thank you, Kamu. Just 30 more seconds. Then we're going to mark. Okay. All right. Wow, this was long, but yeah. Let's do it. Okay, so I need someone to tell me there's 2 strands here. I don't know which one we'll have to use. Anyone to tell me which one we're using? Is it the upper one or the lower one?
The upper one.
Thank
Upper.
you. Thank you. All right, we're gonna do this together. Okay, so I'm going to read out the bases. If I do make a mistake, please do let me know, because that's a long sequence. But yeah, so what is it writing? Okay, so that will be A, T, G, C, C, G, C. Yeah, give me a second. My pen. Okay. Okay, so this will be A, U, G, C, C, G, C. Okay, that's fine. Then I'm trying to keep track here. Then U, yes, yeah. Now I have to start from the first one because I can't keep track. A, U, G, there's the 3, C, C, G, there, G, T, T, C, A, A, there, then U, A. Okay, then it will be C, U, G, then it will be U, U, C, then it will be A, C, G, C, then it will be A, C, U, C, A, U, G, U, G, U. Shoo! I'm not even sure of my bases because that was so long. I couldn't keep the right sequence. I hope I hope it's correct. Is it correct, guys? I messed it up at the beginning, but I fixed it quickly. Is it correct? Thumbs up, response, something.
Yes, it's correct.
Thank you so much. All right, we're skipping these diagrams. There is the information about translation. So, but these are the points you need to mention for you to get 6 marks. Each transfer RNA carries a specific amino acid. What are we describing here? Translation. Each transfer RNA carries a specific, please include the word specific, because it doesn't just carry any amino acid. It carries a particular one. And it will only carry that particular one when the anticodon of transfer RNA matches with the codon of messenger RNA. 3 bases in a messenger RNA codon. 3 bases in a transfer RNA anticodon. Then what will it do? It will then bring the required amino acid to the ribosome. It collects it from the cytoplasm and then bring it to the ribosome. Now, what will happen to these amino acids? They are going to be attached to each other by peptide bonds to form what? To form the required protein. Skipping this. This is all the diagrams. But before we come here, I need you to write down this somewhere on a paper. So we are going to look at the sequence of molecules that are formed. Whenever you are answering a protein synthesis question, I need you to write this down. Where did we start? We started by using DNA. And then from using DNA, what molecule did we form? We formed a messenger RNA. From messenger RNA, what did we form? We used the codons of messenger RNA to form the anticodons of transfer RNA. From the transfer RNA, we are then able to get the sequence of amino acids, because remember the transfer RNA is the one that will collect the specific amino acids and bring them to the ribosome. When we have a sequence minimum 50 amino acids, we then form a protein. This is your guide to protein synthesis. Please do write it down. We started by using a code from DNA to make a messenger RNA strand. We use the codons of a messenger RNA strand to get the anticodons of transfer RNA strands. The transfer RNA then collects the specific amino acids and brings them to the ribosome. Then in the ribosome, that chain of amino acids forms the protein. This was our goal, and we achieve it. Okay, same. I want us to answer this together. So just 2 minutes for you to look at the diagram, and then we answer it together. Just look at the numbers and look at what is happening here. So these are still just basics to understand the diagram. Okay, the first question says we must label 1 to 9. So there is 1, there is 2, there is 3. 3 is this space over here. There is 4, there is 5, there is 6, this part over here, and where is 7? I don't see 7. Oh, there is 7. There is 7 over here. There is 8 over here. And there is 9. Okay. So I need people to answer, guys. Please, if I call out your name, please do answer. Please, as a belief. Okay, Sane, what is 1? There is 1 over here. What is that? What is that label there? Okay, Kanyisa, what is 1?
Ma'am, is that not the nuclear membrane?
Perfect. Nuclear membrane. Remember, this is the nucleus. So that's the nuclear membrane. Perfect. 2, Asanda. Asanda, please give us label 2. Okay, then there's an IP here. So IP, please do give us label 2. Okay, Bongo Kuhle.
Is it the M?
Yes, I'm listening. IP, you were speaking. I don't see the name. It's just written IP.
Oh.
So
Mine needs mRNA.
mRNA, perfect. That is the mRNA. There is number 2. That is the mRNA. Okay. Then 3 is this space over here. This little gap over there. Bongo Kuhle.
Ma'am, isn't it tRNA? I'm not sure.
Let's see. Let's look here. There's number 3. See the space over here? Remember, we are having the membranes. And then we have the spaces between the membranes. Do you remember what we call those spaces?
Nuclear pole.
Nuclear pole. Thank you. And then 4, there is 4. Remember, guys, you also have these diagrams on your notes. Then 4, Luyanda.
Transfer RNA.
Transfer RNA. 5, I will accept 2 options for 5. So 5, Tabo.
Is the protein chain?
Protein chain, I will take it. If someone said polypeptide chain, I will take it. Then 6, Neliswa, 6.
Hold on.
Yeah, I'm hearing someone. 6. Okay, anyone, guys?
Amino acid.
I can't hear.
Amino acid.
Amino acid, 6 is definitely amino acid. Then 7, there is 7 over here. Anyone, please give me the answer.
Ma'am, isn't it codon?
Is it codon or anticodon?
It's
Anticodon.
anticodon.
Anticodon, these are bases in a transfer RNA. Okay, then there is 8 over here. These 3 bases over here. Anyone? I can't hear. Codons. Then 9, this organelle over here. That's 9. What's the name of that organelle?
It's the ribosome.
Thank you. Then we give the names of process A and process B. There is process A and there is process B. Can someone name process A, please?
Transcription.
Process A
Transcription.
is transcription.
Thank you. And then process B.
Translation.
Thank you so much. Let's do this one. Actually, 5 minutes. I'll give you 5 minutes to do this one. Complete, let me break it down for you. Complete the corresponding messenger RNA codons and transfer RNA anticodons for these DNA triplets. So you are given these DNA triplets. We are calling them triplets because we read bases in 3s. Just like we also have codons, 3 bases, anticodons, 3 bases, DNA triplets, 3 bases. So number 1, you're going to complete a messenger RNA and a transfer RNA strand. So you use this DNA to form a messenger RNA first, and then you form a transfer RNA. That is number 1. Number 2, You are going to use the table below. You have it on your notes. You use the codon table to work out the amino acids that are coded for. So let's look at this table. So this is very important for you to know how to interpret. This is the table of amino acids and you get given in a test. So you don't need to cram anything. So this table will normally have the name of the amino acids. And then on this side can either have the DNA triplets or can have the codons or can have the anticodons. You use what you are given. In this table, we are given codons. So that means we are going to use the bases in a messenger RNA to find the name of the amino acid. So that's exactly what we're going to do. Whatever base you have here, the codons you have here, you're going to take those bases. If one of the codons is GGG, you're going to look for GGG here, and there's GGG, and there's the name of the amino acid, and you write it down. Question 3 is asking you, how many amino acids are coded for by this section? 4. How many different amino acids are coded for? Meaning you will have the messenger RNA, transfer RNA, and then you actually need to have the names of the amino acid because that is number 2, then number 3, and number 4 you can answer. 5 minutes, guys. Please do try your best. I can see other people, I'm not sure whether they don't want to talk or they don't know the answer or they are not paying attention. It's quite worrisome. But yeah, I'll give you 5 minutes. I will turn off my camera. I'll go and make coffee or get water and then come back. After 5 minutes, we are marking. Please do try your best. If you don't have the table, please do let me know. I can take a picture and send it to the group. But yeah, now I'm just projecting these questions. You should have the table on your notes, but like I'm saying, if you need me to take a picture and send it to the group, please do say so. 4 minutes left. Time flies. Okay, I'm checking. Are we done or do we need 2 more minutes? Please do respond so that I know what to do, guys.
Two
Are we
more minutes.
done? You need 2 more minutes.
Yes.
Okay, thank you. Let me know when you guys are done so that we can mark. So I'm just here waiting for you. Can we mark or do we still need 1 more minute?
Last minute, ma'am.
Okay. All right. I'm sure you can see that I've started because I also am slow and I'm trying to not make mistakes because you can sometimes make mistakes here. So I'm hoping my bases are also matching with your bases on your side. Now we are going to try and figure out the amino acids. If there's anything wrong with my bases, guys, or a mistake I made somewhere, please do let me know. It can be, you know what, so easy for me to make a mistake because I mean, it's been a long day and I've been teaching since 7 a.m. So I'm going to use now. So that means we've answered number 1. Number 2 says we must use the codon table to work out the amino acids coded for by the sequence. So I'm going to use it part by part. Now the question would be, which bases are we using? Are we using the transfer RNA or the messenger RNA bases? We are guided by the table. The table has given us codons, so that means we are going to use codons, which is in the messenger RNA. Okay, so AUG, that is my first codon. I'm going to look for it. There is AUG. What is the name of the amino acid? MET or START. It's the same thing. So I write it down here, MET, then dash. Then ACU, I go and I look for ACU. ACU, there is ACU. We are given THR. THR. Okay, then the next one, AGC. I'm looking for AGC. There is AGC. The name of the amino acid, SER. SER. Okay, and then the next base, UCG. UCG. I'm not seeing it. Let's see.
It's TRY.
Meaning I made a mistake here. UGG. Sorry, this is my G, not a C. UGG. Where is it? Oh, there. I hope I'm correct, guys. Please, let's see. UGG. Yes. So TRP or TRY. I did hear someone. So TRP or TRY. Doesn't matter which one you use. Okay. Then GGG. I did see GGG just now. GLY. GLY. UAU. UAU. TYR. TYR. Then UAC. UAC. TYR also. TYR. And then UUU. PHE. And then UAG. Stop. All right. Stop. That's what you needed to do for number 2. Are we still on the right track? Yes, ma'am. No, ma'am. Please, something. Yes, ma'am. No, ma'am. Thank you so much.
Yes,
Thank you,
ma'am.
guys. Thank you. Oh, you're making me feel better. I've been sad for more than 30 minutes now. Okay, I need someone to tell me then. Number 3, how many amino acids are coded for in what we have just done? How many amino acids? Anyone?
9.
9.
8.
Ma'am, I got
8.
8.
8.
So there's
Me
8
too,
and there's
Father.
9.
9. 9.
Okay, let's count them.
9.
1, 2, 3,
Oh, it's
4,
nine.
5, 6, 7, 8, 9. How many amino acids are coded for? We are counting the amino acids. We don't check which ones are repeated, which ones are not. We are counting the number of amino acids. Question 4. How many different amino acids? That will be 8. So this will be 9 amino acids that are coded for. How many different? 1, 2, 3, 4, 5, 6. We're not going to count these one also because these two are the same. So 1, 2, 3, 4, 5, 6, 7, 8. There is 8 different amino acids. Yes, I'm hearing someone. I'm hearing an O. Yes, yes.
Ma'am, can I ask a question?
Yes, Kamu.
Ma'am, so do we count for the amino acids till you get to stop or till you get to the PHE only?
Okay, this stop is the name of the amino acid. So we count it because this stop was determined by these bases there. So we do count it.
Oh, okay, ma'am.
Yeah. So I'm assuming the people that said 8 here, maybe they didn't count the amino acid stop. It's actually a name of them, an amino acid, which you are finding from the table. So you do count. So there's 9 amino acids here, physical count. And how many are different? It's actually 8 because these two are the same. There's 8 different amino acids. All right. Okay, let's come to this one. The DNA coding strand sequence is given. So this one, they've told you it is a DNA coding strand. Give the sequence of the messenger RNA codons. I am looking at the time, guys. So this should be simple. Oh, but there's quite a lot. Okay, messenger RNA codons. This will be U. Wow, what a day. Can't be you. I'm trying to erase. Chances are the screen will become blank on your side. Okay, that's fine. A-U-G. Now my eyes are wide open. Then I need to use my other thumb. It'll be A-A-U, and I need to say it out loud. Then A-U-A. Then it becomes U-U-A. Then it becomes G. CA, then it becomes CGC, then UUG. All right, there wasn't enough space for me to just write the bases directly below. Then transfer RNA, I think we can work this out, guys. Transfer RNA, we are going to use the messenger RNA to form transfer RNA. Use the table of anticodons to work out the amino acids. So I think you can do this, guys, at your own spare time. So let's look at what is different with the table. We are no longer using codons like the previous example. We are using anticodons. So that means you first need to find the anticodons. You're going to use these codons here to find your anticodons, and then you find the name of the amino acid. You answer the same thing. How many different, what do you notice about UUA and UUG? You'll notice that they code for the same amino acid. I'm trying to get to a complex question. This is what I want. Okay, our last activity. Please do this one. I think the rest you will do it as homework. Please do this one. Please try your best. There is a table here, and then there are questions. There you go. So I'll give you maybe 4 minutes, 4 to 5. And this one is from a previous question paper. From a previous question paper, so please do try your best. Okay, let's finish, guys. 5 minutes is done. Let's finish, please. The sequence of amino acids, let's finish with a bang, please, assembly. The sequence of amino acids in a protein molecule is coded for by DNA and RNA. The table below shows some messenger RNA codons and the corresponding amino acids. Let's look at this. According to the table, how many codons code for the amino acid phenylalanine? How many codons code for amino acid alanine? Let me see. Yes, I'm listening.
2.
2. Thank you. That's what I like. 2. There's 2. How do we get that 2? There is phenylalanine. There is phenylalanine coded by 2 codons. Give the anticodon for glutamic acid. They want an anticodon for glutamic acid. Who can give me the answer? Anyone? Let's finish with a bang, guys, please.
CUC.
What did you get? I'm listening.
CUC.
CUC. Perfect. That is correct. How do you get it? For other people that didn't get it, please do explain. I didn't see who answered. Please explain how you got CUC.
From G and G. G is it C, A is it U, G is it C.
Perfect. So they wanted you to give an anticodon of glutamic acid. There is glutamic acid. But now this is not the anticodon, that is a codon. So you then need to find basis of the anticodon, which will pair with GAG, which is CUC. Perfect. Then a section of messenger RNA has the following base sequence and is read from left to right. Give the DNA base triplet for the last codon of this section of messenger RNA. Give the DNA base triplet for the last codon of this section of messenger RNA. Anyone?
TGG.
TGG, perfect. TGG, tick. Give the first amino acid coded for by this section of messenger RNA. Give the first amino acid coded for by this section of messenger RNA. Anyone?
Aspartate.
Aspartate. How did you get aspartate?
From the table, man.
Table. Remember they said first amino acid. So we are going to use these 3 bases, the first 3 bases, right? And how do you then get aspartate? So these 3 bases are G, A, U, yes? Okay, then you go to the table, you look for GAU, there is GAU, there is the name of the amino acid. That's it. We are going to leave it there for today. Now, I will resend the activities, the same activities from last week. There was DNA replication and protein synthesis. If you check that document, you will see that those activities are from previous papers, 2024 and 2023. Nothing earlier than that, right? Please do the protein synthesis and those DNA replication questions. I will check if I've sent it on the group, but I think I did last week. If it's not there, I can resend it. In next week's lesson, we are going to mark those activities. For protein synthesis, it won't help if I just send the memo. We are going to mark some of the activities. because they are challenging. There's quite a few of them, so please do find time and do those activities. I will also do mutation to close off the topic. If you saw on the notes, there's a section about mutation, and then there is also activities from the notes. So, however, we're not going to be able to now do all the activities. It's on you to do the activities, and if you need a memo, you then let me know. I send you the memo or I check your work. That's it from my side. I do have a plea from you guys. Next week, let's be more interactive. It is quite draining to be the one speaking all the time, and then when you ask people questions, no one is answering, or the people are not answering that you're asking questions from. This is supposed to be beneficial for you too, so please, please, I'm begging you, next lesson, let's be more interactive. And like I said,